For the next few weeks, we're going to be doing different kinds of ordination. Last time I talked about the difference between constrained and unconstrained ordinations, and today we're going to do one of each. We're first going to perform an unconstrained ordination, PCA, Principal Components Analysis, on a large site-by-species data set to try to identify the most influential sites and species accounting for most of the patterning in the data, resulting in ordination axes that summarize the data along those most important variables or principal components. We will then use those PCA axes as dependent variables in a constrained ordination, redundancy analysis or RDA, to examine the influence of environmental variables from a site-by-environment data set on the distribution of species within sites. So PCA and RDA are both forms of ordination. PCA is unconstrained, meaning that you're looking at only one data set at a time, whereas RDA is constrained, meaning that the patterns in a site-by-species data set are presumably constrained by variables in a different data set, a site-by-environment data set. PCA is typically used to reduce the dimensionality of a large data set of interrelated variables. But I want to stress to you that it does not remove variables, nor does any other form of ordination. It simply summarizes it into the most important groupings of variables that explain the most patterning in your data. And those groupings are the axes, the ordination axes, or the principal components. Then you can use RDA to examine the relationships that environmental variables have on those PCA axes or on those ordination axes, on those most influential sets of variables. And so when performed together, they are a powerful pairing that allow us to determine or try to determine the effects of site, species, and environment. PCA is an unconstrained ordination technique that summarizes data redundancy by identifying the dominant gradients of variation and placing similar sampling entities in proximity to each other along those gradients in ordination space, resulting in a very economical description of the data. To do so, it creates new composite variables that are called principal components from the original variables. These principal components are linear but uncorrelated combinations of the original variables, and these principal components axes represent gradients of maximum variation. These principal components form axes in ordination space, and so when we plot these, they will have like PC1, PC2 as your axes. The first axis will represent the most variation, the second axis, the second most, PC3, the third most, and so on and so forth down the line. And there will be as many axes as you have sites or samples, in other words, rows of your data set. This technique is primarily used as a way of detecting independent combinations of influential variables. So suppose you have samples in environmental space or in species space. What PCA does is that it takes your cloud of data points and it rotates it or arranges it in such a way that the maximum variability is visible to you. This identifies your most important gradients. Usually only the first couple of principal components are important. They're the ones that account for most of the variation in your data. For any form of ordination, or any statistical method really, you need to understand the data assumptions and the limitations of the method. In PCA, the primary data assumption is that there are linear relationships among your variables. In other words, linear relationships between species abundances or presences and the environment. However, this seldom is the case. It's much more likely that species have a unimodal response curve to the environment. That is, species usually peak in abundance at some point on an environmental gradient. It's possible that a short portion of that gradient, there may be a linear response, as you can see with these three hypothetical species. There are short portions where the response is linear, positive or negatively. But for the entire species distribution, there's a unimodal relationship. If you have such a data set, then PCA is probably not going to apply. Or if you suspect that there is a longer environmental gradient, then PCA is not going to be your best choice. What you can do instead is first you can try to transform the data. Try a logarithmic or a power transformation first. But really you ought to consider a different form of ordination. So, for example, correspondence analysis is another type of unconstrained ordination that doesn't have a linear assumption. You can also try some other forms of unconstrained ordination to act as alternatives as PCA. In addition, PCA does not perform well on data sets that have lots of zeros in them. And that is manifested as what's called a horseshoe effect. So you can see this curvature here like a U. In this case, the toe points down, but the toe of the horseshoe could also point up. In this particular example, we have 12 sites numbered sequentially from 1 to 12 to represent positions that they should occupy along an environmental gradient. And PC1 is your horizontal, your x-axis. That's the one that accounts for most of the variation in your data. Then PC2, your vertical axis. And so sites 1 through 12 ought to arrange themselves in order along that axis. And you can see that in this case they do not. That sites 3 and 10 occupy the most extreme positions along the horizontal axis. That's because you have this horseshoe. In a horseshoe effect, the second axis is a curved distortion of the first axis, which makes interpretation difficult. You expect PC1 to be the most important, the most influential axis, but the horseshoe shape confounds this and violates the independence of these axes. The PC axes ought to be orthogonal or independent of each other, but the horseshoe shape violates that. And in this case, the second axis has no easily understood biological meaning. There's no obvious reasons why, if you look at this in a vertical sense, why sites 6, 7, and 8 should be at the opposite ends of a gradient from sites 1 and 2 and 9, 10, 11, 12 kind of thing. So instead of going from sites 1 through 12 on the horizontal axis, as it should, the most extreme sites are 3 and 10. And so in this particular example, it's easy to see the horseshoe effect because there's only one clear gradient and because the gradient is so strong. But in many data sets, there may be more gradients or weaker gradients, as well as more noise, making such plots much more difficult to interpret. If you have such a data set, again, you can try a transformation first, such as a chord or a Hellinger transformation, but you really ought to consider a form of unconstrained ordination that is an alternative to PCA, such as correspondence analysis, which is also called reciprocal averaging. There are 4 components of output of PCA. The first is the percent variance in the data set that is explained by each axis. So each PC axis, PC1 or PCA1, they both mean the same thing. Each axis is an eigenvector. And so in this case, the first 2 eigenvectors, first 2 axes, explain only 47.9% of the total variance of this data set, meaning that most of the variance is coming from factors that you didn't account for or didn't measure. In general, if the first two PC axes don't explain at least 60% of the total variance, then your PCA will not be useful. But in practice, I see this rule of thumb violated all the time. Indeed, it has been said that PCA is not only the most commonly used form of ordination, it is also the most commonly misused form of ordination. So this result isn't great, but I'm going to continue with this example for illustrative purposes. So that's the first thing you look at. Then you're also going to look at species loadings to try to identify which species are the most influential in the data set in terms of accounting for patterning in the data. You're going to look at site scores to determine which sites are the most influential. And then you're going to make ordination plots, particularly byplots, where you're plotting two forms of data simultaneously, the sites and the species. PCA loadings and scores allow you to identify which species and which sites are the ones most responsible for patterns of variance in your data. Loadings are sometimes called component loadings or factor loadings or weights, and they tally the importance of your column variables, your species. So here's an example. And in this particular example, this one indicates that the value of a sample along the first axis of the PCA, PC1, is 0.9688 times the standardized abundance of species 1 plus 0.9701 times the standard abundance of species 2 minus 0.1045 times the standardized abundance of species 3. So we can interpret axis 1 in this case as being positively related to the abundances of species 1 and 2 and weakly negatively related to the abundance of species 3, or associated with the absence, weakly associated with the absence of species 3. Axis 2, on the other hand, over here, is positively related to and therefore correlated with the abundances of all of the species, especially species 3. So the gradient reflected by axis 2 is something that species 3 cues in on. For scores, scores assess the importance of your rows, your samples, or your sites. And in this example here, PC1 is most affected by site 2 and PC2 by site 3 here. The positives and the negatives mean something only within the same PC axis, not across axes. So you look at plus and minus within a principal component rather than across. So you factor each principal component as its own unit. Okay, so this is how you assess the importance of species and sites on your principal components. Okay, so here's an ordination plot, and in this kind of plot, the circles are your sites or your samples. And what you do is you first look at PC1 to see if there's any patterning or separation in the data along that axis. And so in this case, we can kind of see that, yeah, there's this sort of clot of sites on the left, and there's this grouping on the right. So sort of like this, I've indicated by these circles. And then you do the same thing if you see anything on the vertical axis, PC2, and there really isn't any separation of sites clearly on that axis, which isn't surprising because remember that the first axis is supposed to explain the most variation in your data. So this is not a byplot, however. It doesn't include the species on it, and so it's of limited utility. Instead, if you want to know how the species respond to your ordination axes, or how they fall out along it, you need to make a biplot, like this one here. In this plot, whereas before the circles indicated sites, in here it's the site names in text, and so they occupy the same positions in the ordination space, but it's just a little bit messier because they're the site abbreviations. The pink arrows and the names are the species in this, and so you can see with this biplot much more information than just the plot we had before. You can see how certain species are associated with certain sites, and how most of the arrows point to the right of on PC1, and so it indicates that PC1 is associated positively with the abundance of those species. And then there are a few species that where the arrows point to the left, or so that means that PC1 is associated with the absence of those species. So this is how you interpret the ordination plots. So in the online notes, we worked through an example of PCA using the Bryce Canyon vegetation dataset again. And there are a couple of commands that you can use for PCA. Actually, there are three of them. There's PCA, which is from the lab DSV package, and then there's PRComp and PrinComp from Vegan. You can only use PrinComp if you have more sites than species. In other words, more rows of data than you have columns, which is not the case for the Bryce Canyon vegetation, so we didn't use that. In practice, PRComp is used most frequently for PCA analysis in R. When you measure variables in different units, you should use a correlation matrix, because that standardizes the variables to a comparable scale. So, for example, if you have a plant community where you count some species as individuals, like trees, but other species are counted as clones or tussocks or any other circumstance where the number of individuals is estimated rather than counted, like might be the case for grasses, for example, then you need to use a correlation matrix. It can also mean that when you have species with abundances that differ by orders of magnitude. When the variable scales are similar, you can use a covariance matrix. However, in general, it's recommended in community ecology that even if all your species are measured on the same scale, to prevent the dominant species from really overly influencing the results of your ordination, you still should use a correlation matrix. The downside to using a correlation matrix is that it's a pretty brutish way of standardizing your data and can muffle the variance that you're trying to examine. So anyway, we went with the good standard practice of using a correlation matrix, and in that, depending upon whether you use PRComp or PCA, you use a different command to standardize the data. In PRComp, it's scale equals true, and that scales the data by subtracting the mean and dividing by the standard deviation. And in PCA, it's correlation is true. You can also center the data, and centering is done by subtracting the column means, and it is center equals true is the default in PRComp. If you do want to include it, it needs to be specified before your scale command. So here are the three commands, and we used PRComp and PCA as examples in the online notes. Again, you need to evaluate whether you're going to use a correlation matrix or a covariance matrix, but in general, a correlation matrix is the one that's recommended in community ecology. You want to scale or figure out if you want to scale your variables. Now in the notes, we started with scale equals false. So that's because we measured vegetation, it was all measured by percent cover, and that was all the same. So we don't need to scale it. And you wind up with this ugly thing. You can see a very strong horseshoe effect in these data. These are ugly. And that comes from the scaling issue. Horseshoe bad. So if you go back and instead say scale equals true, then you wind up with, yay, something that's more informative and useful. So in PCA, remember that there are 4 pieces of output that we're looking for. First is the percent variance explained by each axis, and hopefully the first 2 axes account for at least 60% of that variance. If it doesn't, then the PCA probably isn't going to be very informative. Then you want to look at the species loadings and the site scores in order to determine which species and sites are the most influential. And then finally, you want to make an ordination plot, especially a byplot that puts both the sites and the species on the same graph for interpretation. So, for the Bryce Canyon vegetation data set, let's go through each of these. First up is the percent variance explained by each axis. And unfortunately, in this case, you can see that the cumulative proportion of variance explained by the first 2 axes is only 47.9%, less than 60%. So really, we ought to stop here and go do something else, but we're going to continue on with this for illustrative purposes. We can plot this variance divided by each, accounted for by each axis. We can graph that in what's called a scree plot here. Remember that you're going to have as many PCs as you have sites or samples, and we have far too many. We have, what, 160 sites? We have far too many to really make a meaningful graph. I've only plotted just the first few because the axes after the 4th one account for a minute fraction of the total variance, and once you get past the 100th axis, then it's almost sitting at 0. It's so tiny. Okay, so what you're looking for in the scree plot is the big drop-off between PC1, 2, and everybody else. If they're all very flat, then PCA is not for you. You should go do something else. Okay, so now we have this. Now, who's accounting for this variance? What species and what sites? So, first up, species loadings. And this is how you interpret this. I've only plotted this for the first 6 species listed by their abbreviations here. Remember that there were 169 plant species. And what you do is you look within each PC axis. The first thing you look at is the absolute magnitude largest values. So in this case, ARCPAT has the largest number here. And so the others are much, much smaller than ARCPAT's number. So in this case, that means that the presence, since this is a positive number, the presence of ARCPAT is strongly associated with PC1, or rather I should say that PC1 is strongly driven by the presence of ARCPAT. It's also driven by the presence, positive association, of this one that has the abbreviation of AMEUTA, and PC1 is affiliated with the absence of the other species, the ones with the negative signs. Now remember that positive and negative signs are only for within each PC axis. Okay, so we've established that PC1 is associated with the presence of ARCPAT. Now let's go to PC2. Again, look at the magnitude, the absolute magnitude of numbers first. And you can see that the biggest number is also with ARCPAT. But in this case, it's a negative sign. And that means that PC2 is most strongly affiliated with the absence of ARCPAT. So PC1 is associated with the abundance of ARCPAT in a positive way, and PC2 is associated with the abundance of ARCPAT in a negative way. So that's how you interpret the species loadings here. Okay, so keep this in mind. PC1, presence of ARCPAT, PC2, absence of ARCPAT. Now let's go on to the site scores. And here you do something similar. You look at each PC at a time. So PC1, first off, you look absolute magnitude, biggest number, and it's site 5, clearly. So PC1 is strongly affiliated with site 5. Then we go to PC2, do something similar. What's the biggest number? Okay, it's site 2. So that means that PC2 is most strongly associated with site 2. Okay, so when you put these two things together, the species loadings and the site scores, in this case, remember that PC1 is strongly associated with site 5, and it was strongly associated with the presence of ARCPAT. PC2 is most closely affiliated with site 2, and it was most strongly affiliated with low abundance, negative abundance, or absence of ARCPAT. So when we go back and we look at the actual veg data itself, you can absolutely see that for ARCPAT. You can see that it is much more abundant at site 5, and it has its lowest abundance at site 2. Okay, so that's what the species loadings and the site scores can get for you. It can help simplify this, because remember that this is a big, hairy 160 by 169 data set here. Okay. Now, and finally, we plot. We produce a plot, an ordination graph, like this byplot. There are other R packages that can produce more attractive graphs, like ggplot, but we're not going to go down that rabbit hole. We're going to use the base plots produced by the packages. In this case, PRComp. PRComp's option for byplot, giving you a byplot, is one reason why it is used more frequently than PCA from LabDSV, which only gives you a plot rather than a byplot option. In this byplot, the site abbreviation, site names, are in black text, and the species are in pink. The arrows are indicative of abundance. Longer arrows indicate species of higher abundances. And you can clearly see how certain species are strongly associated with the conditions found at certain sites. Notice also how most of the arrows point to the right, meaning that they are positively associated with PC1. There are a few species that are negatively associated. If you look at the vertical axis and look at which arrows have a positive slope versus a negative slope, you can see that there are about as many positive associations as there are negative ones with PC2. Okay, so now that we have some useful axes of variation, the principal components, we can now use those axes as dependent variables in other analyses that allow us to examine the influence of environmental variables on species at sites. That means we need to use a constrained form of ordination. Like RDA, or redundancy analysis. Just as with PCA, or any form of ordination, you need to understand the data assumptions and limitations of the method first. Like PCA, RDA assumes that there's going to be a linear relationship among your variables, and therefore it's likely only applicable along short environmental gradients. If you suspect that you have a longer environmental gradient, or that your species have unimodal rather than linear responses to the environment, you can first try to transform your data, or you can use a different method. Just as correspondence analysis was the unimodal analog to PCA, canonical correspondence analysis is the unimodal analog to RDA. Yes, this gets very confusing. There's correspondence analysis, there's canonical correspondence analysis, and those are two different things. CA is an unconstrained ordination, like PCA, and CCA is a constrained ordination, like RDA. We got acronyms and abbreviations all over the place here. Stay focused. Just like with PCA, RDA does not perform well if your data set has lots of zeros. Again, you can try a transformation, like a chord or a Hellinger transformation, or you can try a different method, like CCA. We will cover CCA in an upcoming lesson. A constrained analysis allows us to examine the effects of environmental variables, like those from BryceSite.R, have on the simplified, the ordered or ordinated data that was generated from a PCA. You technically don't even need to do a PCA if you use the RDA command from Vegan. It will do one for you, but its plotting options are not nearly as good, and so I recommend that you go ahead and use PRComp anyway. So in the online notes, we used the RDA command from Vegan, and here are the environmental variables from BryceSite. You can see that some are numeric and some are categorical. RDA only works with numerical data, and so I picked elevation, aspect value, and slope to examine. These data were centered and they were scaled, and then they were plotted. This is a triplot because it plots the sites in circles, the species in red points or red crosses, and the environmental variables in blue arrows. In this resulting triplot, you really can't make out any patterns with respect to species. They form this red smudge in the middle of the plot because the variation in the sites and the environmental variables swamps any patterns with the species out, because the variation in the sites and the environmental variables effectively stretches the plot. Arrows that are parallel to an axis are more strongly associated with that axis, and arrow length indicates correlation strength. Thus, you can see how aspect and slope are negatively associated with each other because they point in opposite directions, and they are both affiliated with axis 2, RDA 2, because they're vertically aligned. RDA is most closely associated with elevation, with increasing elevation. So now if you look at the distribution of the site points, you can see how sites in Bryce Canyon differ more in slope and aspect than they do in elevation, because they're more stretched out along the vertical axis than on the horizontal one. The plants, however, don't seem to be strongly associated with any of these three variables. When we take these results and consider how little variation was explained in the PCA axes, however, it's clear that factors other than elevation, slope, and aspect are at play in structuring the vegetation at Bryce Canyon National Park. So hopefully you can now perceive how PCA and RDA are very similar and quite complementary to each other. PCA is an unconstrained form of ordination. It searches for any variables that best explain species composition, whereas RDA is constrained. It searches for the best explanatory variables, your environmental variables. PCA can help sort a large number of correlated variables to an ordered set, which can then be used in a subsequent RDA or any other form of constrained ordination. Performed sequentially, they are very good at detecting species-environment relationships. And so now let's look at some empirical examples from the primary literature that used PCA and RDA. Eczema is an autoimmune disease that is inflammation of the skin. It is characterized by redness, itching, and embarrassment from unsightly, rough, cracked, and peeling patches of skin. It typically affects children and is also associated with respiratory allergies later in life. It is as yet uncurable, but it can be treated, especially if you catch a flare-up early. In this study by Hong et al., they examined the gut microbial community in infants with and without eczema. They didn't ID the microbes to species. Instead, they had what they called, or what are called, operational taxonomic units, or OTUs, that were based on 97% genetic similarity. Those acted as their species. So first they examined how the microbial community changed over time, and that's in plot A. And then they compared the microbial community in infants with and without eczema, and that's in plot B. In plot A, the PCA was based on the presence and absence of these operational taxonomic units at different time points: 1 month, 3 months, 12 months, and 2 years of age, in children of 2 years of age. The authors concluded that the presence and absence of these microbes were time-mediated. But I ask you to notice that the axes explain collectively only 29.6% of the total variance, far less than the advised 60%. So I would not bet the farm on that result. In plot B, it does better. It at least has at least 60% of the variance explained. In fact, it has more than 90% of the variance explained. So at least that's better. And it's based on the abundance of classified microbes within eczema and non-eczema groups at different time points. And you can see the 1M, 3M, 12M, and 24M, the 1, 3, 12, and 24 month children. And they concluded that PCA indicated that abundances of specific microbes correlated to eczema status during various early stages of infancy. They concluded that these differences indicate that microbes from fecal samples of infants can provide potential biomarkers to distinguish infants with eczema from those children that do not have it. These findings may help understand the possible pathogenesis of certain microbes and may lead to a timely intervention that reduces the occurrences of early life eczema and possible allergic disorders in later life. This study by Ringel-Kulka et al. Similarly looked at the gut microbiome in infants, comparing their gut community with that in adults, because although it is generally believed that a person's microbiota is established in the first 1 to 2 years of life, this hadn't yet been tested. So they examined the microbial community at 2 taxonomic levels, that of the phylum level, shown here, and of genus, which will be the next slide. And it's actually phylum-like and genus-like because of these operational taxonomic units, owing to the difficulty of assigning species identifications to so many microbes. Okay, so as you can see, they did both a PCA on the left, panel A, and an RDA, panel B. In the PCA, the first 2 axes represented only 37% of the total variance, which again is less than the recommended 60%. But these axes were used anyway in an RDA on the right, which show that there are differences in the gut microbial community between children and adults. The authors concluded that the establishment of an adult-like intestinal microbiota thus occurs at a later age than was previously reported. Similarly here, at the genus level, there was the microbiota profiles of children and adults did not separate out at this more refined taxonomic level, unlike the previous graphs at the phylum level. But again, the first two principal components only accounted for 44% of the total variance. However, the RDA of the genus-like level microbiota profiles separated young children from adults here, although not clearly on either of the two axes, really more on tandem. So anyway, these were two examples, this study and the previous one, of how PCA and PCA with RDA have been used and used ill-advisedly. So for your assignment, you're once again going to be working with our friends, the ground beetles, the ground beetle abundance data, and environmental data from the GB site CSV file, and use what you learned today to address these 5 questions. You're going to do a PCA and you're going to do an RDA. And in the PCA, you're going to go through and generate and interpret the output, the percent variance that's explained. Hopefully it will be greater than 60% for the first two PC axes. The loadings and the scores to account for which species and sites are driving most of the variation, the patterning in the data. And then you're going to generate and interpret a byplot of this. And then you're going to do a constrained ordination on this in terms of looking at how vegetation height possibly structures the abundance of beetles at our 18 sites and 3 habitat types that are represented in these two data sets.